Welcome to this news briefing from the 249th National Meeting and Exhibition of the American Chemical Society in Denver. We're joined today by Dr. Laura Kiessling from the University of Wisconsin-Madison. Dr. Kiessling delivered the Fred Kavli Innovations in Chemistry lecture yesterday and has made discoveries in carbohydrates that could help lead us in the fight against antibiotic resistance. Dr. Kiessling?
Thank you. Our research focuses on the glycans, that is the carbohydrates that are on the surface of all cells. So every cell on the planet has a coat of carbohydrates on its outside. And we became interested in the difference between the coats of carbohydrates that are on microbes and those on mammalian cells. So because this carbohydrate coat is an evolutionary conserved feature of cells, it plays important roles in recognition, and in many cases, it's essential for the viability of microbes. So what we were interested in doing is exploiting the fact that microbes use different sugar building blocks than mammals use on their cell surface. And so if microbes have essential carbohydrates composed of different building blocks, we could take advantage of that in order to target them. So I think everyone's aware that there's really a need for new antibiotic targets because of the resistance problem. And the ability to use this difference in carbohydrate coat has not been very well mined at all. So I think there's a lot of opportunities. So we envision, and we've been exploring, two kinds of opportunities. One is to block the biosynthesis of the unique carbohydrates, the carbohydrates unique to the microbes. So if you have inhibitors that block those key enzymes, you could potentially use those inhibitors themselves can function as antimicrobials. The alternative strategy is to use proteins that can selectively bind to the microbial cell surface, including those carbohydrates. And while we haven't been exploring that, that possibility in detail ourselves, we have found a human protein that can selectively recognize the microbial cell surface carbohydrates over the human cell surface carbohydrates.
Thank you. Let's open it up for questions. Please state your name and affiliation before asking your question.
So it's Kath O'Driscoll, it's from Chemistry and Industry magazine. Maybe I could just ask about what are the advantages of targeting carbohydrates over proteins? Would they confer maybe some advantages in terms of, if you were to develop antibiotics, are they more, because they're conserved, would that mean that they may not be develop resistance?
So one of the enzymes that we've been targeting in my lab is an enzyme that's essential for mycobacteria. So mycobacteria cause a number of diseases, but especially and most notably tuberculosis. And they use a unique carbohydrate, galactofuranose, that we don't use. And so so to get back to your question, interestingly, the enzyme we've been studying that is critical for incorporation of that carbohydrate is not, if you look at different strains of mycobacteria, it's very well conserved across strains. And it's also very well conserved within an individual. So if you monitor, so even within an individual, you can have different mycobacterial strains that evolve. And so this is one of the enzymes that varies little. And so that's part of the reason we were interested in it, because it could be more difficult to develop resistance. So I can't say that for every particular enzyme. What I can say is that because those pathways haven't been mined, we don't have a lot of built-in resistance, which you have for some of the drugs.
Could you also say something more about this human protein that you've identified that binds to the glycans, the microglycans?
Yeah, so it's a human protein that's actually produced at mucosal barriers. So it's in your, it's produced in your intestinal epithelium and your lung. And it's been implicated in many properties. So it had been identified as a protein that binds to a microbial carbohydrate, but it had also been implicated as a tumor marker. It is upregulated in diabetes. So, and there are human mutations in this protein associated with Crohn's disease and asthma. So it had this sense that it might be involved in, let's say, defense against microbes, but we still actually don't know the full function of this protein. We just have a lot of circumstantial data. But what our results show pretty clearly is that it does not bind to any human glycans, so it doesn't bind to any of the carbohydrates that are in us, but it does bind to carbohydrates on the surface of microbes. So it's certainly exciting to us because I think it's likely to be involved in defense, but again, we don't know for certain.
Also, are there sugars on the surface of viruses? I mean, could this approach also be perhaps extended to look at viruses?
Yeah, so this protein there could be viruses that it could recognize, but a lot of the viruses, they have carbohydrates made from our mammalian biosynthetic pathways. So it depends on where that virus came from, but probably not. So it probably wouldn't be involved in direct defense against viruses. But that doesn't mean there aren't proteins that have that role.
Okay, thank you.
Ben Valsler, Chemistry World. Given that these appear to be specific for at least families of microbes, if not necessarily strains, does that also mean that this approach will allow us to be quite specific for what we're targeting? So we'll get antibiotics that will kill off an infectious agent but wouldn't harm the gut flora, for example?
I think that is a possibility if you, I think that the issue that you raise is very relevant. And I think in part that's why people haven't looked at some of these biosynthetic pathways, because if you block, let's say, RNA polymerase in microbes, you're going to kill all the microbes. And so we had all these broad spectrum antibiotics. That's what most of our antibiotics are. But now I think people are thinking, because we know so much more about our microbiomes, that it would be good to have more selective antibiotics. And so these would be more selective, the kinds of things that I'm talking about. The interesting thing is that the protein that we were targeting, that I talked about, trying to find inhibitors of this one particular enzyme that's quite invariant across different strains, that enzyme is found in a bunch of other pathogens that you might be interested in targeting. So it's not only found in mycobacteria, it's found in some fungi, and also, especially like Aspergillus, fumigatus. So these are fungi that cause major human diseases. And it's also found in nematodes. So it seemed to us like a really interesting target because, again, all of these too are microbes that afflict the developing world. And so it'd be great to have some tools. And one of the things that I talked about is that we found inhibitors of this protein that do work on different homologs of the enzyme. So we can, we have some molecules now that will kill mycobacteria, but they'll also kill nematodes, working through that same protein. So.
Why do you think it's taken so long to start mining this route? You'd think that a pathway that's found in a broad range of pathogens would have been identified a while ago and would have presented itself as a target very early on.
So one of the ways that people tend to look for antibiotics is it's very effective if you can do whole cell screens instead of target-based screens. And so what we tend to do is either use natural product libraries, extracts, or collections of small molecules. So we've looked at how effective the current collections of small molecules are at targeting proteins of this type, and they're not good. So if you run a high throughput screen, our hit rate is really low. And that's hit rate on an isolated protein. So I would say, if you think about the kinase field, right? We now have all these really amazing selective kinase inhibitors. But, 20, 25 years ago, we didn't. We didn't really have the tools to do that. And it's really been a lot of beautiful and elegant chemistry that's led to these really effective and selective kinase inhibitors. Not just chemistry, right? Also structural biology. And so all I'm saying is if you do a whole cell screen and you don't have compounds that can even hit that target in your library, yeah, you're not going to find compounds that hit those targets. So what you tend to do is find compounds that hit the same old targets, because that's what's in our library. So one of the other goals of our work is to really try to figure out what kind of chemotypes, what kind of molecular scaffolds we can use to target this kind of enzymes in this broad class.
So speaking of the beautiful chemistry that's taken place, could you just take me through what the different steps are? So we have a microbe of interest. Where do we go from there?
So there are two philosophies, right? You could look for inhibitors against particular targets. So what we typically do is ask what genes are essential. And there are techniques like transposon mutagenesis. You can genetically look for what's essential. Once we know those genes that are essential, we could then isolate those proteins and try to find inhibitors. The other way to do it is say, let's just look for things that kill the microbes. And so both of those are very valid ways of going about the problem. And what a lot of the libraries, the kinds of compounds that we have out there, have not been especially good at killing microbes. There was a very famous GSK study that was done where they found all the essential proteins for some, I don't know if it was Strep aureus, but it was some microbe that we wanted to target. And then they made all these proteins and they ran all these assays and they identified potential leads, but none of those really led to new antimicrobial agents. So then that led people to say, well, never do that. You need to screen on whole cells. And so, but often if you screen on whole cells, you identify compounds that hit the same targets. And then if you already have microbes that can take advantage of mutations, you can quickly develop resistance to the compounds that you have. So I would say we need all strategies. We can't, that's my intrinsic personality. I'm not a 1 or a 0 on anything. I think multiple ways need to be explored. And so why the GSK study failed in the first place? Maybe because the compounds that they had were not that permeable across the microbial cell wall. And in fact, that is what a lot of people have found, that the compounds that we have in our collections that maybe have been made to treat, cancers or other sorts of human diseases often aren't permeable to the cell walls of microbes. We haven't been very good at identifying those compounds. Now, those libraries weren't necessarily selected for that property. So I think research on the chemistry side could lead to better compound libraries. And I think, research continuing to look for clever new screens that monitor maybe not just life or death, but some pathway can also lead to new antimicrobial agents. So.
Thank you.
Question in the back?
So we have an online question from Christine Suh, ACS News Service. So she's wondering, what are the next steps that you need to take to develop this enzyme for potential diagnostic or treatment purposes? And I'm also wondering, I'm Katie Cottingham from ACS News Service, like, are you partnered with companies to do this development?
So right now, some of the work I'm alluding to is our best inhibitors have not been published. So we're just getting ready to submit that work. And so my goal, I do think that there are companies that are interested in this. At the same time, there are also agents like the TB Alliance that can help take drugs for mycobacteria, help develop those drugs. Because as you would guess, anti-tuberculosis drugs, that many of the people who die of that are in low-income countries, and so it's not super lucrative. So you just have to find the right partners. The nice thing is that there have been some good mechanisms put in place, and I know the Bill and Melinda Gates Foundation has obviously been trying to develop alliances and start new interactions that can facilitate drugs being, made, antimicrobial drugs getting to market.
All right.
I didn't. No, I don't think I answered the first question though. Do you want? Oh yeah. So. What?
So for this particular enzyme, what are your next steps in order to develop it for potential diagnostic or treatment purposes?
So right now, we just got a structure of our small molecules bound to this enzyme. And so one of the things we want to do is take and leverage that structure to up the potency. Because we have a big boost in potency over what we've published before, but we've done virtually no chemistry on these compounds. So we think a little chemistry and we can up the potency significantly, and then we want to pursue that direction.
Do we have any other questions? If not, I'd like to thank you all for joining us today. The archived version of this session will be posted soon at bit.ly/ACS Live Denver. Please join us for our next press conference today at 2 p.m. on turning fat into potential diabetes treatments. Thank you.